evolutionary development, which is one of the main reasons a lot of people get into evolution in the first place, is they see these changes between organisms and they wonder, how did that happen in the first place? For example, talk about the evolution of flight. We've got two mammals here that are both, well, one's not a powered flyer, one's just a glider, but still. How did these traits evolve? What sort of genetic changes can dispose or predispose an organism to developing these huge flaps between its limbs? Most of us don't do that. So what sort of mutations can cause this, and how likely are they, and what genes are involved? What sorts of mutations are involved that cause these major transitions in development, in how organisms look? And that's the concept of the study of evolution of development. So how do these developmental pathways mutate, change, evolve to lead to differences between, say, chimpanzees and us? What mutations cause us to be less hairy than chimpanzees? Those sorts of things. I'm going to go pretty quickly through a couple of hypotheses about how development evolves. That is, theoretically, and this is sort of the first question on the quiz, not really a quiz, whatever, entry activity. What sort of mutations can cause these sorts of transitions? What do you think is most likely? And then we'll talk about a few cases of actual mechanisms of evolutionary development. So we're going to start with the hypothetical. We're going to follow up with the case studies from scientific literature. We're going to start with this example of the Drosophila mutant Apterus, or Aterus, depending on whether or not you pronounce the P. Anybody know pterodactyl? So, good. What about Archaeopteryx? Yeah. So do you pronounce the P or not? It's from Greek, and in Greek it would be a silent P, so it would be pteryx, or pteris, or pterodactyl. What does that mean? Pteris. Wing. So if you're a wing, the prefix a means you don't have it. So the Apterus mutant of Drosophila is a wingless mutant. So this was identified a long time ago. Somebody was growing Drosophila and discovered there was a mutant fly in their collection that didn't have wings. This is the perfect thing that evolutionary developmental biologists love to find is mutants, because then we can start asking, well, okay, what was the mutation or what were the genetic changes that allowed this sort of trait to evolve, the loss of wings? You can imagine that if you study how wings get lost, you might at the same time learn how wings are gained. What's the gene that's involved? What does it do? What tissues are it expressed in? Is it a structural protein? Is it a transcription factor? What does the gene encode? What does that protein do? All questions that evolutionary developmental biologists ask. By the way, if you just came in, please take the Socrative activity at our usual room number. So these are the goals. So these are the hypotheses. How could developmental change occur? So you have mutations in the part of the gene that causes production of a protein, the coding region. You could have the evolution or mutations in the promoter, changes to the DNA that tell the gene how much it's transcribed or where it's transcribed, which cells, which tissues. Or you could get the evolution of entire genes all at once. And that was the basis for this first question on the quiz, just getting your feedback. Which do you think is most likely to occur? A, the duplication of a gene, B, the deletion of a gene, C, mutation in a regulatory region, which would cause the gene to be turned off or turned on or attenuated, less transcription, more transcription. So you're just changing the amount of the gene that's produced. D, a mutation in a regulatory region that causes the change in what cells turn the gene on. Look, everybody's sleeping, including the computer. Or is it E, a change in the actual DNA sequence of the gene, the coding region? So is it a change in the amino acid sequence of the protein itself that's encoded by the gene? So a few people think that any one of these things occurs. Most of you seem to think that it's a mutation that causes a change in how much a gene is expressed. So you've got a gene that's already being transcribed and translated, and a mutation occurs that either makes more of that protein or less of the protein. It doesn't change anything else. All 5 of these are things that actually happen during the evolution of development. You get gain of genes, loss of genes, mutations that do all sorts of things. And in fact, one of the points I'm going to try to leave you with, and this is one of the critical tenets of evolutionary developmental biology, is that it's often not C that happens. It's very often, it's also not very super, if anything, if any one of these does not happen, it's usually E. So if I was going to rank these, I would say E is the least likely. A change in the actual DNA sequence in the coding region that causes a change in the protein coding sequence. That happens, but not necessarily as frequently as, say, D, which I would say is one of the more frequent causes of the change in development. A mutation in a regulatory region that causes a change in which cells turn that gene on. And we're going to see an example of that in Drosophila. Again, if you just came in, please do this Socrative activity. Any questions at this point? All right. So to restate what I just said, it's very often regulatory DNA sequences that cause a change in development patterns. Can you imagine why this is? You tell a story that changing how much of a gene is expressed or where it's expressed is very much more likely than to cause a change in development than a change in the DNA sequence that causes the protein sequence. So much greater than, so regulatory mutations, much greater than the evolution of protein coding regions. Protein sequence is less likely to change. The amount of the transcript, where it's transcribed, how much it's transcribed is more likely to change. What's the difference? Well, maybe we'll see. So if you flip to the next slide, you'll see a few different examples under each of these bullet points. So protein coding regions evolving, that could be, say, mutating the actual coding sequence of the apterous gene. Regulatory regions evolving might involve altering where or when or how much of the apterous protein is produced, and the evolution of whole genes might mean that you delete the apterous gene. Make a wingless fly, you'd have to know something about how the fly knows where to put its wings. Is anybody here taking Biology 155? Developmental biology. Okay, good. So you've probably not heard this before in quite a while then. I'm sure Dr. Calderon would have told you about this if you had. So there's a group of genes of which apterous is one called the Hox genes. They're the homeobox. Hox stands for homeobox. They're a group of transcription factors. And the really cool thing about the Hox genes, there's a number of them. They all have different names, or they've been given different names. by different scientists. For example, UBX stands for ultrabithorax. They have these very interesting names that say something about, I don't know, ANTP, antennapedia. Has anybody heard of the antennapedia mutant before? What happens when you're an antennapedia mutant?
Antennas become legs.
Yeah, you get pedias on your antennas, or ped, feet. So you get legs. Your legs grow where your antennas should be. Antennapedia mutant. Okay. Ultrabithorax, grow two thoraxes, or you get two thoracic segments. So these are genes are what control, in each segment of the Drosophila, the fruit fly body, they tell that segment of cells which type of cells they are. Are you abdomen? Are you thorax? Are you head? Are you leg? Are you wing? And the really cool thing about the Hox genes, which is sort of incidental, maybe, is that they are oriented on a chromosome in a gene cluster. So all of these genes are right next to each other on one chromosome in Drosophila. And look at their arrangement from left to right, or from 5' to 3' on the chromosome. The genes are located in the order anterior to posterior that they're turned on in the organism itself. So if you have a gene that's up here at the 5' end of this cluster, it's expressed in the head. And the abdominal genes are at the 3' end of the gene cluster, and they happen to be the genes that are turned on in the abdomen, or the abdomen, the posterior end of the fly. So what do you think would happen if you deleted the antennapedia gene? So here's the antennapedia gene. It's expressed here, right, where wings form, where the second set of legs form. What would happen if you just deleted that gene?
Would it only have 4 legs?
You might, I mean, that's a great hypothesis. It might be a fly that just has no wings and 4 legs. It might not develop that segment of its body at all. Whether or not that's lethal is something we could debate. Maybe you don't get a fly at all. But that would be the effect. If you don't have that gene, the Drosophila body doesn't have that segment. There's no gene, this transcription factor doesn't exist. It doesn't get expressed. It can't tell cells in the middle of the fly to become that segment of the fly's body. The really cool thing about the Hox genes is that they're really broadly conserved, meaning that if you have a Hox gene in one species, it's probably expressed about the same time during development and about the same place in the body as the same gene in a different species. This is so much the case that you can actually take an Apterus mutant Drosophila and you can stick in, transgenically, the human version of the same Hox gene. And look what happens to the fly. It grows its wings again. The human version of this gene you can put in a fruit fly, and this human version of the gene works in the exact same way as the Drosophila version of the gene, despite how very, very, very different we are from fruit flies. And presumably this is one of the reasons that duplications or deletions of genes is not one of the ways that development works. Rarely, yes, but generally not so much, because these genes, the Hox genes, are so important in developing the body plan. What happens if you start tinkering with them? Not just deleting or duplicating these genes. What about making changes to the amino acid sequences of these genes? What happens? If you make protein sequence changes at all, it's deleterious. It impacts the ability of these proteins to act as transcription factors and turn on the genes that tell different cells in your body what types of cells to become. So the ultrabithorax mutant. You get rid of that Hox gene, either by point mutations, just individual nucleotide changes that cause amino acid changes that cause the ultrabithorax gene to not work so well, or maybe duplication or insertion of a new gene. Cool, we've made that huge developmental change in one step. Change one gene, now you've got a Drosophila with 2 pairs of wings. Is it good or bad? Do you think it's beneficial or detrimental to the Drosophila?
Both.
I don't think you could be able to tell.
So yeah, maybe you can't tell, go ahead.
Unless you were to actually observe it doing its normal
Okay.
daily functions.
So you'd want to know things like,
Does it get food faster?
does it fly faster? Can it get food faster? Can it fly at all? Is it coordinated, or does it spin in circles when it tries to fly? Does it get recognized by members of its own species as a potential mate, or do female fruit flies all of a sudden think, hey, you're not from my species, and so you lose your fitness altogether? So major changes to Hox genes can cause major developmental transitions, like the addition of a set of wings by tinkering with mutations in the Hox gene ultrabithorax. But at the same time, these are major changes that are occurring. And it's not always major changes in development that's necessary to improve fitness for an organism in its particular environment. So you don't necessarily always want to mutate the Hox genes. Hox mutations do happen. There are more subtle ways to get changes in gene expression, though. So this is not changes in the protein coding regions of genes that change their amino acid sequence, like the ultrabithorax mutant. This is an example of how that more subtle change in development can occur. It's not as drastic, say, as the addition of a whole set of wings. So here's an example of how regulatory mutations to genes can cause the evolution of developmental patterns. So what we're looking at here, and this is, despite the fact that this is a cartoon, this is from real genetic studies of Drosophila. There's a gene called yellow, which for some strange reason causes black pigmentation. And so you have to have some Drosophila species have these black posterior abdomens. A lot of black pigment gets deposited at their very posterior end. And that's because they have a couple of DNA sequences, enhancer sequences, that bind to transcription factors, and those transcription factors then are responsible for turning on the yellow gene in the posterior abdomen. And that yellow gene then produces the black pigment. So don't worry about it being called yellow. It's a historical reason. And in this same fruit fly, you'll notice that there's no pigmentation in the wings. So the question is, how do we get some Drosophila that look like this? So there are different Drosophila species that not only have the black pigment deposited in their abdomen, but also they have wing spots. So it's a subtle developmental change, but it's important for mate recognition, for species identification, and so it's critical for their fitness, despite the fact it's only a little tiny bit of pigment in the wings. To the Drosophila, it's a big deal for fitness. So how does that evolutionary change occur? How did they get pigment also in their wing spots? What does it look like? There was a mutation at one spot of the promoter sequence upstream of yellow, right? One or a few nucleotide changes that causes the appearance of a new transcription factor binding site. And that transcription factor is a transcription factor that's only found, or that transcription factor is only turned on in this little tiny spot in the wing. And so that combination is, now you've got a transcription factor that can bind to and turn on the yellow gene in the tips of the wings. So that transcription factor, this is the critical point, the transcription factor was already being expressed in that tiny little segment of the wing, probably for another reason. So it does something important in wing development, say. But this new mutation now allows it to regulate a gene that it has never before regulated, the yellow gene. So you get transcription factor already in the wing, once you give it a binding site upstream of the yellow protein, now it can turn on the yellow gene in the wingtip, produce pigment, you get wing spots. Likewise, what's happened down here in the third fly on the bottom? It doesn't have wing spots, and what else happened? To its pigmentation compared to the other flies. Yeah, it lost the black pigmentation in its bottom. How did that work? Another mutation. A mutation that removed one of the binding sites that's necessary for transcription of that yellow protein to occur. Right? So there's a mutation there that turns off or blocks transcription of yellow in the abdomen. So single nucleotide changes, not in where the protein is encoded, but in the regulatory sequences, can make some subtle changes, but significant changes to development. It doesn't have to be mutations of the gene itself. It could be regulatory sequences that cause a change in how much of the protein is produced, where it's produced, or where it's not produced. Now, it turns out gene duplications happen as well. And the Hox genes, despite being so critical and fundamental to the patterning of our body plans from early in development, they duplicate quite a bit. That whole set of genes, that whole gene cluster, the Hox gene cluster that we see in Drosophila, all in one segment of the chromosome, one after the other. When we look back in time, so to speak, I know this is bad evolutionary terminology, we see that once symmetry started to evolve, our ancient ancestors, the lancelets, have one set of those Hox genes. And that establishes the anterior-posterior axis and body plan of those ancient ancestors of ours. But then when you start looking at closer and closer relatives to humans, you see something interesting, which is that that whole set, that whole block of Hox genes has been copied and pasted into our genome, so that Agnatha, or Anatha, jawless fish, have more than 3 sets of that group of Hox genes. Fish have 4. Well, cartilaginous fish have 4. Bony fish have 8. Same set of genes, copied and pasted 7 times. You've got 8 total sets of Hox genes. We have 4 tetrapods down at the bottom. What's the point of having so many versions of this set of genes? What do you think? Is it beneficial? Is it detrimental? This gets back to the other Socratic question. We've had a lot of gene duplication of the Hox genes going on. Good, bad, or indifferent? Okay, so it could depend in part on where these genes have landed on chromosomes. You copy and paste, and maybe where you paste those extra genes is important. Let's assume for the moment that because we have these extra sets of genes, we're okay with where they went in the genome. So the insertion of this set of genes didn't mutate another gene and cause us to lose things.
Maybe it has to do with if there is a bad mutation that occurs in one set of these Hox genes, that the other set can accommodate for it.
Okay, so it's backup, redundancy. So it could be useful to have multiple sets of Hox genes because they're so important. And if a mutation happened in one, you'd be screwed. But if you've got a backup set of the Hox genes elsewhere in your genome, then if one of the sets of genes, say abdominal A, gets mutated, instead of not developing an abdomen, your other copy of the abdominal A gene gets turned on from the second set of the Hox genes.
Could it also have to do with cell regulation, like the different cell types that different organisms have? So maybe something simpler, something that wasn't even like symmetrical, didn't have as many complicated cells that would need that much regulation.
So it could be, so it seems like there's a correlation between the number of sets of Hox genes and body plan. So maybe complexity evolves with the number of Hox genes, which maybe suggests what about Hox genes? Why would it be useful to have multiple sets? I think you're heading in the right direction.
They're regulating what gets expressed in the cells, I think.
Right.
And so, if you have a more complicated body plan, you need, I guess, more directors to conduct the show.
Okay, so if you've got more Hox genes, then maybe you can make more different patterns, or more different cell types, or more complex patterns. What would that suggest about, so say you're a teleos, a bony fish, you've got 8 versions of the same gene. Sorry, I can't even count. 8 different versions of the same gene. With your hypothesis then, what would that mean about the 8 different versions of the same gene? Are they identical?
I mean, some maybe, I can't say they're
Sure,
not,
yeah.
but I would think that not all of them are. I
Right.
think some of them over time have mutated.
Okay, so if you're going to make a complex body type, and that's because you've got 8 different versions of the same gene, maybe they're all very similar to each other, but maybe some of them have a few distinct mutations that allow that complex pattern to arise. If they were all exactly identical, there wouldn't be any change, presumably. You just have 8 different copies of the exact same gene.
I also going back to what you said about the cell type too. It's almost like coding on a computer, it's all just ones and zeros, but the amount of those ones and zeros and the combination of those ones and zeros get you different outcomes.
Okay, so, yeah, exactly. It's an interesting analogy. I never thought about it that way. I like it. So this is basically what those Hox duplications look like. We have one region that has some of the Hox genes, and then they form tandem duplicates. So all of these Hox genes tend to be in the same region of the same chromosome. They're just head to tail. Copy, paste, copy, paste, copy, paste. All in one region of one chromosome. And that's what we have in our bodies that tell us how to develop in early development. They lay down the initial body plan. And if you actually look at, and this is a cartoon again from real data though, it's just an easier way to summarize all the data. When you look at, say, mouse versus zebrafish, mice have 4, as we just saw, tetrapods have 4 sets of Hox genes. Fish have 8, bony fish. And that's because the fish have undergone a duplication of the set of 4 genes. That's how they got 8. So there's been duplication after duplication after duplication. There's one set, they duplicated to make 2, that set of 2 duplicated and you got 4, and that set of 4 duplicated again in bony fish only. So they have 8 sets of Hox genes. And then this becomes a real issue for the geneticists because you have to come up with some sort of nomenclature to talk about which of the 8 different versions of ultrabithorax are you talking about. So that's where this Hox cluster naming system, which is unimportant to memorize. I just wanted you to be aware of how to interpret this graph. So each of these Hox clusters has a different name. So there's AA, AB, AC, and so forth. Or sorry, CA, CB, and then there's D. Now wait a second, what happened to the 8th cluster? And what happened to a lot of the genes that are in here? Notice that the D cluster doesn't have a lot of the Hox genes in it. Right? It doesn't have group 1 down here on the x-axis. It doesn't have group 1, group 2, 5, 6, 7, 8. So what happened to all these genes that are missing from bony fish? Should have been there. They were originally duplicated. This whole set of Hox genes. What do you think happened?
And they're replicating, they couldn't, because there were too many, maybe they couldn't do it in time.
So there was some issue maybe with replication. The DNA is still there though. So there's still the same amount of DNA.
There's just no expression.
There's just the genes aren't turned on. So this is a sign that there were mutations that caused those genes to become pseudogenes. So everywhere there's a gap here, there was initially a gene, but what happened to it? Etc., and I could do this forever. Plainly stated, each of these genes picked up a mutation that caused it to cause a premature termination codon. It makes a protein that's not functional as a Hox gene. They were subsequently deleted. And generally, we call these pseudogenes. They were genes once upon a time, but they've accumulated a number of mutations that mean that there's basically no functional protein produced from these genes anymore. And that's direct evidence of one of your predictions, which is that you've got a lot of copies of a gene. You don't need them all. They're backups for each other. So if you have 8 different copies of the same gene, how many do you really need to be a functional organism? Well, if you're a diploid too. You only need one of these 8 different copies of this Hox gene to be functional. We don't need all the rest. They're backups, and this is exactly evidence of that. We've got a number of these genes. Most of them in fish have mutated. Are there any of these where there is no functional copy left? Like which one of these paralogs down here on the x-axis, which one of these paralogs is in the worst shape? 1 through 13. Which one's in the most dire straits? Yeah, Hox gene 7, right? It's got 1 left. So mutations have occurred in all of the copies of that gene. There's still 1 though. There's still 1 left. Bad things would happen if a mutation happened in that last copy, presumably. And we can skip this, just a duplicate slide. Any questions at this point? So Hox gene duplications presumably provide a backup for some really critical developmental genes, that if a mutation happened in the 1 copy that you've got in your genome, that would lead to certain death for your lineage, at least, if not for you as an individual.
So with these duplications, is it kind of like the idea of like Mueller's ratchet, where along with good mutations, you're also going to have accumulation of deleterious mutations? And so that's why over time you get some that are losing a lot of the genes?
So it's tenuously connected to Muller's ratchet in that once you have, this is very tenuously connected to Muller's ratchet, but you're right in the concept that once these mutations happen, it's unlikely that there will be a reversion mutation that fixes the initial problem. That's a separate process than Muller's ratchet. But, yeah. And there's so many mutations that can break a gene. It's relatively easy. Once one of those really rare mutations happens, what's the probability of the reversion mutation that reverses the problem happens again? It's even more rare. Is there a detriment to having so many copies of the Hox genes? Is there anything bad about it?
Well, if the one that has the least amount of genes get expressed, then it's bad.
Oh, maybe. Is it energetically inefficient to have so many copies of this set of genes? You've got to copy all of the genes every time you replicate your genome. So could this ever be selected against? Is basically the question.
There's probably a limit.
There's probably a limit. There's probably a balance. So there's trade-off. There's probably some benefit to having some extra Hox genes around just in case you get a mutation in one. But at the same time, yeah, every cell division you have to copy a little bit extra DNA during DNA synthesis. But presumably, because all of these organisms have extra Hox genes, there's some sort of a benefit to having them. So I want to close now. We've already talked a little bit about some of these concepts. I want to close with what happens when you have duplicate genes in your genome. And this is based on theory. There are 4 different fates. So after duplication, you can get one of basically 4 things happen: neofunctiontalization, redundancy, subfunctionalization, and neofunctionalization. I'm going to walk briefly through an analogy of what each of these mean. But you start with, say, our ancestor. 1 set of Hox genes becomes 2 sets of Hox genes. These are 4 theoretical different things that have been shown to happen in different sets of organisms. Neofunctionalization, nonfunctionalization, subfunctionalization, and redundancy that can happen. So we're going to look at each of these 4 terms and what it means to a gene. And by the way, if you came in late, please do this Socrativity. So here's my analogy for neofunctionalization. You've got 2 versions of a gene. You just need one to be effective. So in this case, for our generation or your generation, our generations, let's talk about a cell phone as an analogy for a gene. So it does something that's useful for you, like helps you chat with friends. When you've got 2, what can you do? You've still got one you can use as a phone, you can still text, you can still surf the web, so what do you do with the second one? So one of the things we're going to see is you can keep it in case one breaks, but you've also got a new tool. You've got something else, and you could use it for whatever you want, like a doorstop. That's neofunctionalization. The original copy of the gene keeps its original function. Now evolution has something to tinker with. So mutations can happen in that second version of the gene. Sometimes those mutations cause that gene to break its function. That is, it becomes functionless. Pseudogene, that's a different term. We're going to look at it in a minute. But this is the case where you've got substrate now, you've got a second copy of a gene. It can evolve a new function. It can do something new for the organism. Light be expressed in a different tissue. So the important concept in all of these is the original gene keeps its original function. The new gene does one of 4 different things. Here it enables something new to happen. So that's neofunctionalization. So say you've got a gene that does something pretty complex, like a cell phone. So it's both a phone and a computer. What happens when you have a duplication and you've got 2 cell phones? Another thing that can happen is that the mutations can occur. But in this case, the one mutation to your cell phone causes the telephone to break. So the microphone doesn't work anymore. So that just becomes a computer. Right? The telephone part doesn't work, but it's still a computer. You can still use it to text. And in the other copy of the gene, maybe the computer part breaks, but it still works as a telephone. So now you had 2 telephones that were computers. Now you've got 1 telephone and 1 computer. That's subfunctionalization. So you had one gene that was complex. It did multiple things. After you've got a duplication, one of those 2 tasks can break in one of the genes by mutation, and the other task could become disabled in the other copy of the gene by mutation. Does this impact the organism? Does it still have enough genes to do all of the original functions? Yeah, it's got 2 copies of the gene, but together, each of them separately do both of the jobs. So you're no worse off than you were, you just have an extra copy of the gene. And that's why this is called subfunctionalization. You take the job of one gene, break it up into 2 different genes. Non-functionalization is just the redundancy idea. You've got 2 copies of a gene. If the mutation happens to one that destroys its function, you're fine because you still got the backup copy. So what's the 4th thing that can happen to a duplicate gene? Redundancy. You got an extra phone, that's it. You got that backup copy in case at some point in time one of those processes occurs. Neofunctionalization, subfunctionalization, if the gene function breaks. So this is what's happened to all of these different things have happened to all of the Hox genes at some point. Some of the Hox genes have subfunctionalized. It doesn't matter which ones, I'm just saying in general, all of these things happen to duplicate genes. And this is one of the reasons that evolutionary development biologists have found that over time, gene duplication is often one of the mechanisms, might be the leading mechanism, of how evolution changes and how development changes. You get duplication of genes, then any one of these four processes can work. Most often, in the case of evolutionary development, that's neofunctionalization. A mutation occurs in one of the copies that gives it a new function. That's how new developmental change occurs. Like the wing spots in Drosophila. A mutation that causes a transcription factor change in one of the copies of the Hox genes, and that makes new pigment in the wingtips. You get wing spots. Any questions about this? So, simple concept, gene duplications occur, then one of these four patterns occurs subsequently. And all of these gene duplications give evolution the substrate to work with. Extra copies of genes that random mutations can occur to. Sometimes those mutations do something beneficial, sometimes detrimental. But in the end, because those mutations are occurring in copies of a gene, it's fine for the organism if some of these random mutations occur. So drastic changes don't often occur in the evolution of development. It's often subtle changes, usually to the expression of a gene, where it's turned on, where it's transcribed, how much it's transcribed, not usually in the actual amino acid sequence of the gene. That's how evolution happens. Any questions? All right. Congratulations to you all. It's your second to last 8 a.m. class. You can decide whether or not you want to wake up early and come to the review session on Wednesday. It's the same as every other review session. Come ask questions. And I know it's pretty far in advance of the exam, so you probably haven't started thinking about studying for the final yet, but if you have, please come and ask questions. I'll be here and at your disposal to talk. We've got office hours today from noon to 1:30, and again Wednesday, and that's the last office hour, and I'll be around Thursday and Friday for consultation. We'll see you on Wednesday, I hope.